My name is Yimeng Lyu. I'm a PhD candidate at Georgia Institute of Technology. Today, I'll talk about the investigation of surface reactions during methane activation over nickel oxide and zirconia catalysts using In-Situ IR Spectroscopy. So, a brief background on methane. So, methane is the major component of natural gas. The proven reserves of methane is found to be around 460 trillion cubic feet by the end of 2017. However, due to limitations such as difficulty in transportation from remote locations and its low energy density by volume, such vast reserve of methane has not yet been utilized efficiently. Oftentimes, natural gas and byproducts are often just vented or flared on site. As you can see in the satellite picture here, the Bacon oil field is almost as bright as the city of Minneapolis. The challenges in using methane as a platform molecules lies in first, the traditional steam reforming processes requires large facilities for this process to be economically feasible. And for other possible reaction pathways, the difficulty in methane activation due to its high CH bond energy impedes the complete of the catalytic cycle. So now we're going to explore the few possibilities there for reaction pathways of methane valorization processes. So first we can have the non-oxidative coupling, which gives us aromatics or higher alkanes. So these reactions are selective, however, often suffers from the thermodynamic limitation and can only be operated at high temperatures. To remedy that, we might want to introduce oxidants. We can have the oxidative coupling of methane, which produces olefins, and olefins can be further upgraded to other products too. We can also have the reforming-based routes, which produces first syngas, and then from syngas we can either convert the syngas to higher alkanes through Fischer-Tropsch synthesis or methanol through methanol synthesis. However, these processes are two-step high-temperature and pressure processes, which requires extra energy input. So ideally, we want to have a direct partial oxidation route, which produces methanol using cheap oxidants such as molecular oxygen or water. However, these partial oxidation reactions often faces the fierce competition from the combustion of methane to CO2, and hence it's challenged by the selectivity limit. And finally, we can always just burn out methane and gives us energy, but this produces CO2, which is a major greenhouse gas. So, to address all these challenges, our group has designed this nickel oxide or zirconia catalyst, which has been found to be active for various methane valorization reactions. So the idea behind this catalytic system is that we want to use loose acidic nickel oxide, which can activate methane, and we want to use synthesis to control our nickel particle size to be as small and well dispersed as possible, as such species are believed to improve the activity. On the support side, we chose the redox active zirconia as it activates the oxidants as well as facilitates the methane activation. And additionally, zirconia is believed to stabilize small nickel oxide clusters. So now we know this catalyst is active for methane activation. We would like to learn how the methane activation is actually happening on these catalyst surfaces. So to do that, we're going to use this experimental tool shown here, which is a in-situ IR cell. So here, so in the cell here, we can actually press our catalyst into the wafer and place it into the middle of the cell, and then flow reactants over the catalyst. We can also heat the cell to desired temperature, which makes the cell essentially a mini reactor. So as the reaction happens, we can use the IR beam to probe whatever surface species formed on the catalyst. And in the meantime, we can monitor the products using online GC or MS. So in summary, in this study, we're going to use the In-Situ IR Spectroscopy here to investigate the methane activation over our catalyst surface. All right, so now we know what catalyst we're going to use. We're going to actually go down the synthesis and the characterization of our materials. So this is actually from our previous study where we screening through several synthesis methods, and we found the combustion synthesis to be the most interesting. So the procedure is basically here. We first dissolve the metal precursors, nitride metal precursors, in water, in solution of water, and then add citric acid. So in this system, the nitride in the precursors are oxidants, and the citric acid are the fuel. So then we're gonna just heat this solution. So upon heating, the water will first evaporate, and once the water is fully evaporated, we're gonna see the citric acid gonna polymerize and form a greenish resin. And we're just gonna keep heating this resin. Upon additional heating, just keep in mind that we have an oxidant and the fuel in the system. So upon further heating, this resin will just combust. Will consume all the foreign material that we brought into the system and leaves us our combustion synthesis catalyst, which essentially is a mixed oxide of nickel, serum, zirconium. We can always adjust the weight percent of the nickel or of the nickel that we loaded into our catalyst. So in this study, we're going to use the 2 weight percent and 10 weight percent of nickel oxide over serum zirconia as our test materials. So now we have our catalyst. We're going to perform some characterization. So in the middle, we have the X-softs of our catalyst. Using X-soft feeding, we can actually estimate the nickel particle size. And as you can see that well-dispersed nickel species were observed in both catalysts. If you look at X-softs, we actually see, especially in the 2CS spectrum, we're actually missing the nickel oxygen nickel shell in that sample. So this indicates that we actually have isolated nickel species in this material spectrum. The other characterization we performed was the temperature program reduction. So comparing the three curves here, we can apparently see that the incorporation of nickel into the zirconia increased its reducibility, and the reduced nickel during this reduction process facilitates the activation of hydrogen, which leads to the earlier reduction of the zirconia support in our catalysts. So one additional thing is that the first peaks in the two catalysts were assigned to the adsorbed surface oxygen, which we believe were quite active, and in this previous study we've shown here that is actually responsible for the removal of coke accumulation on the surface during methane dry reform. All right, so now we have synthesized our materials and we have characterized them. We actually want to go down to the experiment, that is our methane activation examination using In-Situ IR Spectroscopy. So the experiments we're going to run is we're going to test the two materials we've just shown at different temperatures by flowing 20 SCCM of methane over the cell, over the catalyst. And then we're going to observe whatever surface species form on the surface during this during the experiment period. So this data set shown here is actually collected using the 10 CI sample at 200 degrees Celsius. So already we are seeing various surface species form during the activation of methane, which is quite exciting. However, to fully understand this data set, we definitely need more detailed data analysis, as well as obviously the peaks are quite overlapping and convoluted here. So the goal in data analysis step would be we want to identify and quantify the growth of different surface species. All right, so as shown in the previous slide, this data is quite complex. So to achieve the goal, we're gonna try to find a rigorous way of doing that. So here we've developed this normal data analysis method which uses linear regression to actually review the evolution of different surface species over time. So the first step of our method will be we want to identify the surface species through the convoluting difference between spectra at adjacent times to multiple peaks. So something like this. So here I'm showing the difference between the 10th minute of the experiment and the 20th minute of the experiment. So you might be asking, why are we fitting the differences? So the reason is that if we have a newly formed surface species at a later time, that is actually overshadowed by the already accumulated surface species. If we just fit that at using the overall data, those newly formed peaks might not be obvious. So by analyzing the difference between different time stamps, that allows us to review any tiny features that are actually on the surface through the deconvoluting process. So the next step will be we basically perform this deconvoluting process over all the the time labels that we have, and we're just gonna look at each data set, each time label and grouped peaks that shares the similar peak position and variance at different times together. So by doing that, this peak is now a representation to a certain species, and it is actually characterized by the peak shape, peak position, and the variance. And then we're just gonna find each and every of the unique peak group of peaks that is out there and put them together, and we will establish a vector space. So this vector space actually contains all the possible surface species that can form over the experiment period. And the next step would be we want to identify each species from either literature or reference experiments. So here is an example of the vector space that we form. So at this point, these peaks don't have physical meanings, but we will show how we're gonna identify these peaks later. All right, so now we have the vector space. We're just gonna use this vector space to form linear regression fits to all the data that we collected at different times, and we're gonna find the coefficient for each surface species at different time. So this is an example of the curve fitting using the vector space. So now we know what are the possible surface species that can form during the experiment period, and we know the coefficient of each species at different times. That means we have already resolved the evolution of different surface species over time. So now we're gonna assign we're gonna review the physical meaning of different peaks, and then we're gonna analyze this evolution of different species and try to learn stuff about this methane activation process over our catalyst. Okay, so identifying the surface species. We can definitely look into the literature on assigning peaks to different surface species, but a more straightforward way would be we just dose different molecules on our surface and then compare it to what we're seeing during the methane activation. So here we're basically dosing 3 probe molecules that are the C1 oxygenates. So what we can learn on the CH stretching side, obviously we are seeing characteristic peaks from the formic acid. And we're definitely seeing some contribution from the other oxygenates, but it's not that obvious. So on the right-hand side, the shoulder here around 1585 is apparently the CO double bound from the formaldehyde aldehyde species, and we also assign these sharp peaks to the formates and carbonates that form during the experiment. The later peak around 1060 is actually the alcohol CO stretching vibration, and my colleague Jennifer actually have dosed the aromatics such as benzene, phenol, catechol on our catalyst, and we believe these broad features are actually from the contributions of various aromatics or polyaromatics, and this peak around the 1200 ish is the phenolates related species. All right, so now we know what these peaks means. We're just gonna plot the peaks that represents the same species in the same plot and analyze the evolution over time. So the starting point or the basic case we're gonna use will be the methane activation behavior on 2CS at 200 degrees Celsius. So as shown here, we can see that it's actually sort of three stages going on during the activation. The first stage will be the first 5 minutes, where we see fast accumulation of all species. And then between 5 to 60 minutes, we see a slower growth of formates, but a stable growth of the aromatics and the oxygenates. As you can see, the growth of these species was not disturbed. And finally, in a later time region, when time's longer than an hour, we see that the growth for oxygenates actually stopped, but slower and continuous growth for the other species were observed. So what we can learn from these observations, so I guess, is that we're definitely seeing a change of surface properties. So one thing would be we have the consumption of the oxygen that is stored in the catalytic surface. So we believe that could explain the changes between different stages. So initially we have absorbed easily reducible oxygen, which is quite active, and that actually boosted the formation of formates and carbonates-like species, which are essentially precursors to CO2. And once these are depleted, we might have some contribution from the surface oxygen that's stored in support, which facilitates further formation of these species, but at a lower rate. Similar to the oxygenates, the active adsorbed oxygen are capable of facilitating the formation of these species, but later on, once these are consumed, we are no longer seeing the accumulation of oxygenates. So one other possibility on the surface change would be the coverage of active sites, as we're seeing with forming aromatics. From the accumulation of aromatics and polyaromatics, we might be covering the active sites, and which it could be a reason why we're seeing this lower in the rates of formation of the aromatics in a later time. All right, so now we have some understanding on our methane activation behavior. We want to make some comparisons between different samples and temperatures. So first, we're going to see the effect of temperature. So right now, we are comparing the effect of temperature on the 2CS sample. A few observations we can make would be we see a faster growth of all species at the initial stage, that is less than 3 minutes when temperature is raised to 300. And we also see this interesting trend of the consumption of the formates, aromatics, and carbonates between 3 and 10 minutes that it was not observed at 200 degrees Celsius. And finally, at higher temperatures, we're seeing this continuous accumulation of aromatics only, but the continuous growth for the formates and the carbonates, all the oxygenates, was not observed at higher temperature. So from these observations, I guess a few things we can learn would be, first, the formation of the aromatics were favored at higher temperature. And the second, the formation of oxygenates probably use different sites other than the other species. As we can see, we don't see this consumption phase that is observed for the other surface species. So that probably means it behaves, this different behavior of the evolution of the oxygenates probably indicates this reaction pathway to the oxygenates is catalyzed by a different site. Okay, so now we're going to finally look at the effect of nickel content. So here we're comparing at 200 degrees Celsius with different nickel load. So the first observation we made would be we see a fast growth of formates and carbonates over the 10 CS at the initial state compared to the 2 CS. And we're also seeing fast growth of aromatics at the initial state, but we don't see this consumption of the consumption stage as we've seen in the formates and carbonates in the 10CS sample. And finally, the formation of the oxygenates seems to be independent of the nickel loading when comparing the 2CS and 10CS sample. So that's quite intriguing. I guess what we can learn will be first, the consumption of surface oxygens definitely alters the surface reactions, especially in the 10CS case. The accumulated formates and carbonates were consumed, especially in the 10CS case later on when these active oxygens are depleted. And then I guess we can argue that the formation of the aromatics will favor on 10CS, which is not surprising as those catalysts does possess larger nickel oxide clusters, which should favor the formation of heavier molecules. And finally, given that this oxygenates was independent, the formation of oxygenates was independent of the nickel content, we probably could argue that by the inclusion of the larger nickel clusters in the 10CS did not alter the formation of these oxygenates, I guess, and that could imply that the active sites that actually catalyze the formation of the oxygenates are actually the small and well-dispersed species that were already present in the 2CS sample. All right, so for conclusions, we learned that methane activation can be achieved over nickel oxide or zirconia catalyst systems prepared using combustion synthesis. And we definitely seen various surface carbonaceous species formed, including formates, carbonates, aromatics, and oxygenates from methane activation process. And we've developed this novel linear regression curve fitting algorithm to review the evolution of different surface species. And a few things we can learn from the analysis of our data set would be the oxygen stored in the catalyst actually facilitated the methane activation. And the formation of the aromatics was favored at high temperatures and in catalysts with higher nickel loadings. And finally, we would hypothesize that this well-dispersed or potentially atomically dispersed nickel oxide clusters were likely to be the active sites for the formation of oxygenates. So finally, I would like to thank the support from my advisor, Dr. Sievers, and my colleagues in the Dr. Sievers group. I would like to thank the contributions from my undergrad researchers and collaborators, Dr. Elliot Stavitsky and Dr. Ray Xu. And finally, I'd like to thank the generous funding and support from the U.S. Department of Energy. And with that, I would like to thank for your attention and welcome any questions.
Recording.